Parabéns. Ok, gente, vamos recomeçar aqui. Bem, agora são 10 para os quatro, né? Não está mais na hora de começar. Estamos pensando, né, de fazer uma coisa depois da última aula, amanhã de noite, uma pizzaria, ou assim, né, em algum lugar. Eu mesmo no lugar que eu poderia ir, e dar uma palestra de noite para a Universidade de São Francisco, lá super bonita. Mas a gente precisa organizar isso aqui, ok? Depois a gente conversa mais aqui. Sorry. Tem um bichinho aqui.
Aí atrás um teste só.
So, next large group of forces shaping genetic diversity, natural selection. As I mentioned, in directional ways. In the case of drift, we saw that it took little frequencies all over the place. Now we will have a clear direction. Unless we'll see that sometimes drift melts everything. So we talked about some basic concepts. This is really classical, and it's not until recently that we have started seeing many examples. For many years, the only example we had was for balancing selection and the polymorphisms that protect malaria. But now, of course, we have tons of negative selection and quite a few of positive selection. And we'll see how selection interacts with drift. And we'll see a brief overview of methods for detecting selection, because tomorrow we will go over some of those again. And finally, we will see the pathway or the usual pipeline that's followed from the first description to of the footprints of selection in the genome to the end, to really understanding why is this being selected. And you'll see that's not easy. That's not easy to get a sense of purpose, which is something a little bit tricky, even as almost as a philosophical concept. Still, natural selection, Darwin features the general idea that populations have genetic variation. We know that. We've been talking about sleeps and CNPs and so on. That the environment presents challenges, but that not all individuals respond in the same way to those challenges. That leads that in a sense, who gets old enough to reproduce and who reproduces and who can rear their children, these are concepts that are not completely random, but may be related to genetic diversity. And thus, if there's something that increases the chances of survival, then it will be found at a larger frequency in the next generation. The other way around is something that detrimentally it will be wiped out. we talk about selection and genetic diversity, but selection sees phenotypes. Selection doesn't care about whether you have an I or a G in that particular position. Selection cares about what are the results of that genetic variation. Selection cares about the size of your beak and the shape of your beak. We mentioned yesterday the selection on a zinc transporter. Selection cares about whether you are getting enough zinc. So this means that the effects on a particular site, on a particular locus, on a particular variant are mediated or are as strong as how determinant that variant is in shaping a particular phenotype. For instance, I just said that there are thousands of variants contributing to height. In some cases, height may be adaptive, even being taller or shorter in some cases. Then the selective pressure will tend to be spread over many different loci, and it may be difficult to find the traces of natural selection spread over those many genes. On the contrary, there might be traits that are basically monogenic, and then the connection between selection, adaptation, phenotype, and genotype will be much more direct. There are related to the phenotype. There are different modes of natural selection. There's stabilizing selection that reduces phenotype diversity, in which there's an optimum for the phenotype, and phenotypes at the extremes of those distributions don't do that well and are lost in the next generation. We have directional selection, in which the optimum is in one extreme of the distribution. Here, for instance, here darker snails have more offspring, so directional selection will shift in the following generations toward dark snails. Or we have diversifying selection that favors exceptionally the extremes of the distribution. And then, if you're in the middle, you won't have as many children as you would have if you are lighter or darker. So these are different relations between selection and phenotype. In terms of genotypes, this is translated into positive selection, called Darwinian directional selection, and it refers to the effects of positive adaptation of a particular variant that is being adapted. It's can imagine, for instance, a variant of the immune system that gives better protection against some infectious disease that will be selected for in the next generation. On the contrary, we have purifying selection that affects many de novo mutations that don't do anything particularly good, that touch things that cannot be touched, and that are in different degrees and at different speeds they tend to be eliminated from the population. And we have balancing selection, in which in which two or more distinct phenotypes are produced simultaneously, and the variability in population increases. This is not the same as overdominance. Variant of balancing selection is overdominance, in which heterozygotes are favored and homozygotes are not. But there are other types of diversifying selection. There's, for instance, frequency-dependent selection, in which, and this is quite typical of immune variants, in which it's the frequency itself that has an optimum. It's not necessarily a particular genotype. In the sense that that it may happen that if an allele becomes more frequent, then it becomes detrimental, because a new pathogen will then evolve to attack those individuals. So we will see cycles of ups and downs of that particular selection, and that may not be related to whether these alleles are homozygous or heterozygous, just the frequency in the general population, and how many individuals are then immune.
Which is the red queen, right?
Yes, yes. So this is quite an old review. It's almost 10 years old. Now we have more examples, and of the growing body of evidence for adaptive alleles in different types of conditions or situations. Like, we know that where are you? Yeah. That, for instance, these spots region that is found at high frequencies in the Inuit may be related to marine diet and climate, like these adaptation in Siberians. There's lactose tolerance, which has been selected at different. There's been conversion evolution here. There are different alleles that cause lactose tolerance in East Africa and in Europe. And of course, the usual explanation is that lactose tolerance provides a new source of nutrition in milk and dairy products. This is, but I suspect that the story is much more complex than that. Because you may have some gastric discomfort if you are not lactose tolerant and drink milk. But death is more discomfort, is important than that. So sometimes, if you are starving, even if your belly aches, you will drink milk. So probably a little bit more complex situation.
Because we have to take into account microbiome.
Yes.
Many lactose intolerant people and still tolerant because of the bacilli.
I discovered that I was lactose intolerant not because anything to do with how I digested milk, but because 23andMe told me. Because I knew when I got my genetic results.
Yes, and I am an opposite case. I should not be intolerant genetically. I know my genotype, but I am intolerant because the microbiome has a high importance, and I have irritable bowel syndrome. I'm still drinking diet.
So, besides the classical examples for malaria, we know some other new examples in all the tropical regions beyond the classically described in Africa. There are other adaptations to cholera resistance for cholera resistance. There's a big question mark still about any adaptive alleles to the Black Plague that decimated 1/3 of the European population in the Middle Ages. That's really important. Light pigmentation in relation to latitude and sun. That's also a little bit complex. Growth stature might mean it might not be selected for. It might be a sort of an epiphenomenon. There might be something else going on here. You know that it's particularly the case for women that they stop growing when they become fertile. If you are in a situation of the life traits or life history that it's better to start reproducing earlier, anything that helps you reproduce earlier will be selected for, even if the price to pay is becoming shorter. So this is an example. This is an example of that sometimes it is difficult to understand what is being selected and the meaning of selection. And we will come back to that at the end of the lecture. Yes. Yes. What's being selected and what's just byproduct of the action of those genes. So a little bit of modeling here. And the point is, again, is the same. We do have models. We know how allele frequencies change in response to selection. We can then do cool stuff like estimating the selective advantage of one particular allele. So we can write this. W is the fitness, and we can assign a fitness to each genotype. This is a model. This is not theoretical yet, and compared to the best genotype, and that we will assign a fitness value of 1 to the best adapted genotype. Or we can write a selection coefficient as the complementary to the fitness as 1 minus double. So, and it's we can add now another parameter h, which is the dominance here. It's the dominance coefficient. So if h equals 0, we have a recessive trait in which only the homozygotes are selected for and against. If h is 1, then the homozygotes and the heterozygotes have the same adaptive value, and anything in between. An h of 0.5 is an additive model in which the effects for the homozygotes for the heterozygotes are perfectly halfway between one homozygote phenotype and the other homozygote phenotype. S is really low. At most 3% for positive traits. 3% means that 100 people without that genotype will have, say, 100 children, and 100 people with that phenotype, with that genotype, will have 103 children surviving to reproductive age. So it's not that something magically transforms you into a superhero. We are all mutants, but we are not X-men or X-women. But it's negative all the way down to -1, because of course there are mutations that are incompatible with life. And here are some of the equations. my point here is that I want you to know that they exist. I don't want you to memorize them. Not at this point, not in the 1 hour class. If this were 50-hour population study course, of course, you would have to memorize them, and we would have exercises that we would be tested on them. Just my point here is know that they exist, and here's how Jonathan Preacher explains this. This is the initial frequency. This is the fitness effects, and these are the frequencies after selection. And we have to multiply by the effect and divide by the total of the allele frequencies, because notice that here we have selection against these homozygous, and we have fewer individuals of those. And as a way of normalizing the result of having relative frequencies related to the total of the population, here we have this average W, this average fitness of the population. And so we can model the increase in how the frequencies change, and what is the value, what's the difference between the next value and the previous one, and they make sense, these equations. Look, for instance, if S equals 0, there's no selection, there's no change, because this 0 here multiplies by 0 everything, and the change then 0. If S is positive, then everything here is everything else is positive as well. So then we have an increase. So and if S is negative, we have an increase.
Yeah, it's something.
Yeah.
Regarding the former slide, you were talking about as not the all other ones. Yes, at most 3%. But
yes.
It's important to know that it's in humans.
Yes, of course.
And this has something to do with our reproductive biology, of course.
Yeah.
Since it takes a long time till we
but it's
finally get pregnant, and then we have one child at each pregnancy.
Yeah.
So as I can imagine that the more K is the biology of a species, the higher can be X, because you have more offspring, and so you can test mutations.
Yeah, I was thinking now that you would probably have much higher values in artificial selection.
Artificial selection.
In selection in crops or in domestic animals.
Okay.
In
bacteria, for example.
Or in bacteria. But
and
just
artificial selection for antibiotics.
Yeah, you can imagine that of course if there's a mutant that creates a coat color in dogs and cats that is favored by the breeders, then that's then the S coefficient for that is almost 100%, because only you will treat only the cats or dogs with that coat color.
Even if the coat color goes on with being deaf or partly maybe blind or.
Yeah, that's
in nature, that phenotype wouldn't really resist.
Yeah.
Okay.
Yeah. Wild cats are all the same color.
Mm-hmm.
So we see here the increase in frequency from almost 0 to fixation for different types of coefficients. So the coefficients, if it goes down quite quickly. With a starting frequency of 0.2, we reach fixation, or obviously much faster. And the shape of the curve has to do with the dominance coefficient. It's much faster for the dominant selection, because then more individuals are exposed to the action of selection. Whereas for recessive selection, it takes quite a long time to really pick up the pace. I'm sorry that this didn't come up that well, but we have here you can use this program to simulate drift and selection, and to use that to so you can input here selection coefficient of plus 0.5% and an effective population size of 500. Here it's the same, just with an effective population size of 10,000. So we are having here both the effect of drift and of selection. And by here the blue line, I don't know if you can see, there's a blue curve that would represent the model for an infinite population size, no drift. This is what's supposed to happen if there was no drift. And this is what's happening in a small population of effective population size of 500. It goes all over the place. It may be it's like that, but it's but look here, with an effective population size of 10,000, all these random things, all these random trajectories are much closer to the expected blue curve. There's a mistake here. My bad. It's again 500, not 10,000. Now a negative coefficient. It should go down, but it stays all over the place. And here, even in one of the simulations here, there are in two simulations, we have this detrimental mutation that has become fixed. And now, since selection is relative, now everyone is maladapted. There's no way of going back. Everyone is as bad as one another, so there isn't this differential. So this may happen in small populations, that you can have a relaxation of natural selection. The effects of drift may overcome the trajectory here of a slightly deleterious mutation. Whereas in an effective, with a larger effective population size, everyone dies.
One question. There 10,000 there and there. So I think
This is a mistake. This is 500. And so yeah, you can play around with the program. It's really easy to use. Just plug in the different parameters, and you get these curves. And it's really instructional. You really see the effects. Most new mutations are lost, either to drift or to selection. And we see the action of selection because here in the rare category, this is the drive value count. This is the so-called side frequency spectrum. So we have a proportion of singletons here, of alleles that have only been once, and this proportion is higher for non-synonymous SNPs than for synonymous SNPs than for non-covalent SNPs. This means that natural selection is keeping this variation at low frequencies. And it's keeping variation at low frequencies, obviously, with a stronger force for those that are for those things that actually do something, that vote for something. But rarely, this is sort of a neutralist paradigm. An allele will be favored by selection and will increase in frequency. And then interesting things happen when an allele increases rapidly in frequency. A new allele appears in a given background. This mutation, this new mutation that is favorable, has happened in a certain haplotype. And now, not only this allele will increase in frequency, this allele is linked to this haplotype, and it's the entire haplotype that will increase in frequency. Of course, recombination will start acting and will erode this haplotype, starting from the ends. But still, with the selective sweep in progress, the selection is still acting, is still favoring it until fixation. And then, with a long time, then recombination will again shuffle everything, and we won't recognize that it was a full haplotype that had hitchhiked right with this selective mutation. And with time, we will start seeing new variants that are appearing by mutation. So, two things are happening here. You can see two different things. First, linkage disequilibrium increases. Since it's a full haplotype that is being coming up in frequency, now all of these variants, all of these blue variants go together. This one, and this one. We see them together with population. So linkage disequilibrium increases. Haplotypes are longer compared to neutral variants. And also, since this is increasing in frequency, not only the ancestral allele is lost, we are losing also the alleles that are in the haplotypes linked to the rest of the haplotypes. Where here we have, I don't know, 10, 15 different states. We will end up with probably just 4. The red one and these 3 blue ones. So,
yes, something
yeah.
again. What would be the ideal size of a haplotype that is showing selective sweep?
It's always it's relative. We always we'll see in a minute how we test these and how we turn this for a test for selection.
Okay.
And we do it always in relative terms, but because these effects can be quite local, because recombination can act at different rates in different parts of the genome. So that it's we tend not to use absolute lengths, but how longer are the haplotypes carrying the red variant compared to the haplotypes not carrying the red variant. Yeah. How many times they are longer than the ancestral haplotypes.
Okay.
So, off the top of my head, I would say that you tend to see haplotypes that are 100 kb long.
100 kb.
Yes. So we see in a region with low recombination rates, this will persist. It will be more intense. So we see this drop in heterozygosity. That is more marked when we have low recombination. Compare the blue line here with the blue line here. At 0.1%, the frequency is not increasing fast enough to see this effect. Yeah, or maybe we just see it in the it isn't sweeping up such a long haplotype. Now, over dominance. when we have lastly that the best adapted phenotype is a heterozygote compared to both homozygotes, it can be easily shown that we have an equilibrium frequency, which is in this case it's the selection coefficient for one homozygote divided over the selection coefficient over both selection coefficients, and we have these trajectories that always end up at the same point. Again, this would be in an infinite population. When there is mass, they will be all over the place, and in different simulations, we may end up in different points. Classical example, we need to explain hemoglobin S and malaria and how the heterozygotes are protected against malaria. The homozygote ancestral variant are not protected against malaria, but the price you pay for the protection in malaria is paid by someone else, by the homozygotes for the S allele who suffer sickle cell syndrome. Now,
may I comment again on malaria? I worked on malaria during my PhD, and there are so many different variants that in a way are increasing resistance against malaria. And this is, in my opinion, a case of convergent evolution, because all of them, or most of them, change their birth site in a way that the early forms of plasmodium cannot hide really into the red cell. And this all the red cells, because they should be looked differently, they are easily phagocytosed by macrophage. They're in the deep endothelial system of the spleen. So this is for G6PD, this is for HBC, and also another variant of hemoglobin.
Yeah, it's
So
you get the same phenotype.
Overleukocytemia and reticulosis, cytosis, it's really.
So we are on a fishing expedition now to find these examples of adaptation, and there are many different methods that are relevant, that are appropriate for different scales of the action of evolution. For instance, we can compare species, and we compare species. We'll explain that in a minute with the AULA. And we can compare populations. We can use this general measure of differentiation, so that if we find an allele that has a SNP that has a really high FST, that may have been caused by selection. And we can work within a population and with methods that are based on the site frequency spectrum or in the haplotype extension. Although there's a variance of haplotype extension method that's compared between populations. So this is more or less the same. In the interspecific variation, we will work with this sort of explaining what I meant by site frequency spectrum. We will see low diversity and excess of rare alleles as a consequence of hitchhiking, an excess of high frequency derived alleles. And this is all of the same. And we can work, and we will see an example, the PBS test that works with local genealogies. And these are the different time scales. So if we work with the proportional functional changes, this goes up to we compare them with frequencies, and it's in the millions of years. Heterozygosity of alleles, that can affect the pool of humankind. High frequency derived alleles, population differences, that's more recent. Even lengths of haplotypes tend to be that tends to be preserved at relatively short time scales in the order of tens of thousands of years. So basically, again, many of the mutations that will change the amino acid sequence of a protein will tend to be deleterious. And we see these that we can count for a gene how many of the states we observe are synonymous or not synonymous. And given that we know the genetic code, we can ask, well, what would be the expected number of one versus the other in terms of I can change this A to a C or G or a T. Maybe if we change the A to the C, it's the same amino acid. So I can count this, and I can have an expectation of the sites themselves that before being mutated are synonymous or not synonymous. So this is the KAK-base ratio, also known as the N of S or W, A for non-synonymous, S for synonymous. So if the proportion of mutated non-synonymous sites over synonymous sites below 1 is indicative of purifying, negative for sterilizing selection. It's a pressure to stay the same. That really the changes in amino acid sequence are not being tolerated, and we see fewer than we would expect, given the ratio at which synonymous variation is being accumulated. In a perfect neutral evolution with no selection pressure whatsoever, it would be irrelevant whether a site is synonymous or not synonymous, and they would accumulate at the same rate. This is rarely seen. And if the synonymous, non-synonymous sites are mutating faster than the synonymous sites, this is an indicative of positive selection. There's a selective pressure to change, to accumulate new amino acid changes. And this is our exceptional cases. Of course, this is very limited, just because we're looking like, please, we have this tunnel vision, because it's so easy to predict what is the functional consequence of a mutation in a calling sequence in terms of amino acid change. Of course, there might be regulatory mutations that have large phenotypic impacts, but those are much more difficult to predict or model. So, when we compare humans and chimpanzees, or mice and rats, the genome-wide averages of 4K-AKS are well below 1. The genome-wide averages are about 0.2 in human and chimpanzees, about 0.14 between mice and rats. So, most of our proteins are perfectly adjust machines. You don't tamper with them or tamper with them at your own peril. So much so that when we see KS values of 0.6, 0.7, we get very excited, because we say, even though we are seeing fewer non-synonymous changes than we should see under a neutral model, we are excited because, this is well below the genome-wide average. These are examples of the positive selection, the MPS ratio, and it's there are look, half the list is related to immunity. This is the arms race against pathogens. We have an advantage over us is that they mutate faster, and they can. And we are again in the red-green situation. We are moving constantly to stay ahead of our pathogens. And there's another arms race, the reproductive arms race. In many cases, in many polygamous species, we have sperm competition. The sperm that's better at really getting and fertilizing the egg, well, that's a sperm that's contributing to the next generation. And that in many species, there's an arms race happening quite literally within females, the body of females. If they have multiple partners, then some will win.
So this is why we have the protein P1 at the list.
So there's also some changes in the autocrine receptors. Now, related to the same frequency spectrum, that is, are we seeing a neutral size frequency spectrum? Are we seeing low frequency, intermediate frequency, and high frequency alleles at the frequencies we would expect under neutrality? And I will defer a deeper explanation to exactly the same class because it's much easier to understand once we know about coalescence. But there's the famous legitimacy, and I will pass the opportunity to mention Montserrat Aguadet, who was my evolution professor at the University of Barcelona.
Oh.
Yeah.
Aguadet?
Yes. Yes, and who was a classmate of Elena's mother, and it's all connected. It's a small world. And the French like particularly the fool's test, because fool means crazy in French.
Ah, but he's French?
No.
Oh.
The fool's test. And for local genetic adaptation, we can use tests based on the FST distribution and PBS, the population branch statistic. Here we see an example of the DAFI-O frequency, which is almost entirely restricted to Africa. And we know that, again, it's protective against virus malaria. So, for instance, if this is again, I don't know if this might be Duffy also. This is the distribution of FST values of this measure of difference between populations, and the mean would be here, the median in person would be here, and these values over there at the tail of the distribution. So that's an indication of an allele that's been that has a population differences that are much higher than we would expect based on the rest of the genome. Or here we don't have FST, we have the raw allele frequencies in Tibetans compared to Han Chinese. Each dot here is a particular state. And they are relatively, most of them are along this diagonal line because these populations are closely related to one another. But we have two slips here in the FS1 gene that have alleles at a much higher frequency in Tibetans than in the Han Chinese. And these two SNPs are related to, I think they have something to do with hematopoiesis, because they are really good candidates for adaptation to altitude. And then, if you have population A and B, and there's an outlying population C, imagine 2 European populations versus 1 African population. Most of the genome will look like this. They are close to one another, but the C and they have relatively short branches. And then in some locus, the tree would look like this. The A branch is really long. This A branch may be then related to some positive adaptation in population A. And these are the Greenland Inuit versus the Han Chinese, and here's another group. They have a sample of Europeans. The genome average looks like this, and top signals in these metabolic genes looks like this. The long branch this is the visualization of how faster these genes have evolved in the Greenland Inuit compared to Chinese and Europeans. And this, of course, this is a footprint of natural selection.
So these genes, the FAD S1,
Yes.
2, and S3, they reflect adaptation to the food.
The F. I don't remember what they are, but I remember that F and A stand for fatty acid. So they have to do with fat metabolism. The Greenland Inuit traditional diet is basically seal blubber, seal fat, and that's why they of course the Danish were okay. These guys are not getting heart attacks. What's protecting them?
What's protecting them? And changing the machine.
So now, on the second linkages equilibrium, I will go a little fast here. Here we have the frequency of each dot is an allele. Here on the x-axis we have frequency, and on the y-axis we have measures that could be either linkages equilibrium nearby in a region or homozygosity. So in neutral conditions, it has this curve, because over time, if an allele grows in frequency just by drift, then this is a relatively slow process compared to selection, and there's time for recombination to eat up on linkage equilibrium. But from time to time, we will have outliers in this position that have much higher linkage equilibrium compared to what you would expect given the frequency. And this is happening to young alleles that have been shrunk to high frequency and that are still presenting this long linkage equilibrium. And I won't go into the details of these two of different tests based on haplotype lengths. I will just mention that this XPEHH is stands for cross-population extended haplotype homozygosity. So we can do this within a population for alleles that have been selected in one population, or we can compare different populations and see that they have been adapted in one population, but not in the other one. And yeah, but I will definitely mention the inventor of this test. I introduce Paris Sabetti, who's an extraordinary individual, lots of different talents, and he works at Harvard, and he's also a talented musician, and he had to overcome a really nasty accident that left her almost half paralyzed. So it's really an inspiring woman. Skip. Now, this is more important. These different methods, and for phase 1, we can go on a fishing expedition on the whole genome looking for signals of the footprints of natural selection. And we can use these different methods and have our Manhattan plot of selection across the gene. And we will get a peak. We can then go deeper into that peak, on that specific genome region or on that population, and see which genes are there, and try to restrict it to now zone, and then use in silico methods to predict functionality. The most basic in silico method is just whether a particular variant is totally non-synonymous. But there are other ways that use, for instance, conservation of a species. If something is preserved in different branches of the tree of life, it must be doing something important for the preservation of life. And of course that's also relevant in the prediction also of the pathogenicity of new variants that appear in patients. So yeah, these are. So actually, this means that natural selection, the evolution, the knowledge of evolution saves lives because of this biomedical application. Then we can go even deeper and test for function in vitro, and we can be perfectly sure that this variant is changing the function of this gene in that particular direction. And still, we may not understand why it has been selected. Remember the example of height versus age and puberty? We'll now go through this checklist with one particular example, and at the end I'll have other examples here. So, whole genome scan, we just saw one with PBS, and they got these three signals. Sometimes it's a little bit suspicious that they labeled this peak, and hey, there's this interesting gene, and this peak is almost as tall as this one. There's no label there. This means usually that there's no good candidate gene that they couldn't imagine. Because quite often the limit is the human imagination. They couldn't imagine what was being selected here.
This was done with microarrays? 2015.
Yes. So we have this calcium receptor gene that has a particular variant that's found at higher frequencies, that will be the blue one, in hunter-gathering populations in Southeast Asia. We met first the actors in the Philippines, but there are others also in Indonesia, in Malaysia, and in the Andaman Islands, in the Bay of Bengal that belong to India. You may remember. There was in the news this image of a population in an island in Andaman, that was being conducted for the first time, and they were throwing spears at the helicopter. Was this huge, really recent conflict. All of these populations, besides sharing the lifestyle, hunting and gunning lifestyle, share also a phenotype, the so-called lots of inverted quarters pygmy phenotype. They have dark skin, they are short, they have really curly kinky hair. And here. So, this in this gene, we are now arriving at the in silico methods. There's this CAT score that takes into account basically both biochemical properties of proteins and conservation across species. Gave a value of 18.42, which means that it's maybe anti-disruptive to the function of this protein. So, but still, what was it doing? So it's a good candidate for experimental follow-up, and you can press a key in your computer and get the CAT score. But what comes next is really years and years of work, because what comes next is breeding knock-in mice, that is changing this particular base position in the genome of mice and compare mice with the ancestral and the derived variant. And after years of frustrating work, also in the middle of the pandemic, having to really fearing that the animal facility staff couldn't go and feed these mice because they wouldn't be allowed. Anyway, pretty long story short, the knock-in mice with the human selected variant were heavier and fatter, were bigger animals with a higher proportion of fat in them. So, what makes them all bingo, eureka? We have a function and we have an explanation for the adaptation. Being fat, being able to accumulate more fat might be beneficial for these populations who live, who don't have regular source. They don't produce foods by answering. Here I have a little asterisk. I will come to that in a minute. There was another explanation. An adaptation that most mammals have, including humans, is a direct relation between the proportion of fat in the body and the menstrual cycles. You need having children is dangerous business for a woman's body, and there's also quite some literature on life history, on life histories, on is it advantages for the species to reproduce more or for the individuals to reproduce more, leaving your genes behind that maybe die in the process, or maybe if the nutritional times are not so good, it's best not to get pregnant then and wait for the times. So that's why there's clearly this adaptive mechanism in mammals in which there's some way in which the body senses like the fat content in the body, and if it's probably something happening in the hypothalamus axis or whatever. It's if you go beyond certain limits, then the reproductive cycles are stopped. But the contrary is also true. If you have more fat in your body, you may be able to have children earlier. It will not be it will not be adaptive just by itself. It may be adaptive just as a way of being more fertile. There's another example that's something that really extraction that
I'm sorry, I have a question. Usually in animal models, you use males to offer tests. And so my question is if the results that you showed on the previous slides are males or females? Because you are hypothesizing on a female hypothesis, right?
Yes.
The first
well, here's the real win. The authors of the paper had both male and female mice.
and both
And both,
and
yes. Because it's it would be an additional layer of complexity for the genome, but for the genomic architecture of this, that this patterning would be restricted to females. Males here don't matter. It's all right if they are fatter or leaner. It's the effect on the females that's adaptive.
if I'm not wrong, and maybe my memory could trick me, but usually fatter male breasts are let's say they have a bigger capacity for sterility, so they're not so able, they're unable to get females pregnant or something like this.
Well, that's for the next 5 years of experiments.
Okay.
It's really not easy to explain.
Yeah.
But the functional experiments have shown that the fatter the mice or rats,
Mice.
mice, the more fertile are the females of these mice, or not. This is still a hypothesis to be tested.
It's a general observation in mammals.
Other general But they
I know of an example in deer.
Mhm. It makes sense.
Yeah.
Makes sense. I only wanted to be sure if they evaluate statistically.
There was an example I actually think fast about deer, and how this sort of translates the general nutritional status, and these life history balances about staying alive versus getting pregnant. So, last example in this section, the energy. Incredibly strong adaptation signal in East Asians. When you look at the phenotypes, the energy at the beginning, what we knew is that makes your hair thicker and smoother. It makes you look like a Japanese woman. Now they, you know, those expensive Japanese hair strengthening treatments where you have the perfect Japanese hair. Well, that's come from the other gene. What's adaptive in that? At the beginning, there was the explanation about the hair in the nostrils, and these that these may have been first in Northeast Asia, and it helped to warm the cold Siberian air. But then it was discovered that air is also involved in mammary gland density. here we are getting closer to differences in survival. Maybe this has to be with lactation. So here's another point. It's not the same thing, understanding function and understanding selection and understanding adaptation. I don't think it was, yes, it was the last slide. More questions?
I think it's also very interesting in on the nutritionist point of view that the content of the milk is also dependent on many things. So the quality of the milk itself may change according to the energy.
Maybe this is already solved, but but at the point I didn't follow it further, but the beginning was fine.
Yeah, why?
Yeah. So home stretch now on 20 slides, promise no more, to try and understand some concepts about coalescence and how we have gotten our way of thinking in evolution and population genetics. We have turned it upside down, you'll see.
Because it's structure. I had an experience with malaria, and the gene I was working with, an off-spine vaccine, it has a high frequency worldwide of deleterious alleles that reduce basically reduce the amount of the protein in serum. And this protein is very important in innate immune response. And then I found out, but my supervisor didn't allow me to dig further. I found that the older were the children, African children, the higher was the proportion, this was statistically significant, the higher was the proportion of children with deleterious alleles. And we know from epidemiological studies, epidemiology in general, from malaria, that so around 6 years of age is a threshold of dying from malaria. And in my opinion, this could be maybe some positive selection for, let's say, like this deleterious alleles, but maybe that they will have also a balance in selection.
Okay. So here is some quotes that someone dug up to describe coalescence, and it was repeated in Richard's work. It's not I read Serenity art quite so often. So it says life can only be understood backwards, but it must be lived forwards.
I love it.
And now we are going to understand life backwards. So we'll talk about genetic genealogies and the coalescence, some basic equations for 2 alleles. We will extend that to k alleles. We will find the TMRCA, the time to the most common ancestor. We will see how changes in effective population size change the shape of these genetic genealogies. We will add mutation on top of that. And we will then end up deriving the basis for the genus theory, which is the most usual side frequencies spectrum test. So let's start. And that we share segments of our genome, relatives, but at the end we are all related to each other. There is such thing as a Mormons would like to know, the human genealogy. So in the if we understand if we go forward in time, what is the right feature for models for those genetics that we saw in for the case of drift, we see here each row is a population at a given time, and you see that this individual has 2 children, this has 1, this has 2, this one has none. And in the end, we end up with this sample. So this sample here has a genealogy forward in time embedded to it. They have this common ancestor. That's the common ancestor of all of them at that point. Actually, but the most recent common ancestor would be this one because he or she is the ancestor of all of them as well. Oh, and this, yeah, and this. But if we go backward in time, we start with the whole sample and consider only the sample and go backward in time. So we start in the present and go back in the past in a way in which our sample will coalesce at the point in which they have a common ancestor. And in the end, we'll arrive at the most recent common ancestor. In the most simple formulation, we will start with n sequences of a DNA region for a population, not with whole genomes. We will ignore recombination. It should work for the otherwise. But anyway, we assume no recombination between sequences. So this and this should be little n sequences are connected by a single familiar phylogenetic tree of genealogy, where the root is the most recent common ancestor. And this is only done in genealogical terms. We are ignoring for the first, at least for the first half of the class, what's the actual content of these genome regions. We are not building a phylogeny as we would do normally between species or within species by comparing the actual sequences. This is just a genealogy. We go here. These 2 red sequences have a common ancestor, but we don't care whether because they come from this great grandparent, this great grandfather, but we don't care whether they are actually identical. Whether we don't know about that, we don't know because they carry the same sequence. We just know the genealogy, or we model as we knew the genealogy. So we start with a sample of genes, of alleles in my sample, with little n alleles, alleles in the sense not of different alleles, but of the half of a genotype. And we have, but in the population, I have twice the effective population size number of alleles. This is the sample, this is the whole population. If this red allele has this ancestor in the previous generation, what is the probability that this other red allele has also the same allele, the same ancestor in the previous generation? There are two any alleles, and it has to be this one. So the probability is 1 over 2NE. There's a probability 1 over 2NE. That's 2 particular alleles in the diploid locus coalesce in the previous generation. We will start this piece by piece. Don't worry, we will, in the end, we will start getting meaningful results, but we need the little bits of pieces to construct this jigsaw puzzle. So, as we often do in probability, we will need this probability that the probability that two diploid alleles do not coalesce in the previous generation is 1 minus 1 over 2ne. We just discovered that this is the probability 1 over 2ne. That's two alleles coalesce in the previous generation. The probability that they do not coalesce is 1 minus that. So then we can find a cool result. We can calculate what's the probability that two diploid alleles coalesce exactly t generations ago. We need t minus 1 generation of not coalescing, not coalescing, not coalescing, and then 1 generation of coalescence. So, and this is the exact same formula of another probability distribution called the geometric distribution, and we know that the expectation for the time of that is 2ne. So the expected time to the most present common ancestor for two particular alleles is 2ne generations. The average time that we will have to wait for two alleles to coalesce in the same ancestor is 2ne, which is the probability, and this is probably a leap of faith. Believe me, this is later. This is the case with the geometric distribution. What about k lineages? The coalescence probability for any pairs of alleles is 1 over 2ne, and the technical time to coalescence is 2ne. But now, if we have k alleles, we have k times k minus 1 over 2 possible pairs of alleles. You know these novellas in which, over seasons, every character has been involved romantically with any other character. So this is the number of pairs you can form of characters in a telenovela. K times k minus 1 over 2. So we now have this possible number of alleles. So we would see coalescence there faster, because we are not there demanding that these two, that exactly this one and this one coalesce is any of these 6. Now it may be this, or this and that, and we have now k times k minus 1 over 2 possibilities. So coalescence will go now k times k minus 1 over 2 times faster. So the expected time for the coalescence is of k alleles to form just to arrive at the common ancestor is this 4ne over k times k minus 1. Going forward, funny thing. Half of the time of the coalescence, we will be waiting for the last one to happen. It goes faster at the beginning. We have here 15 possible combinations. Now here we're left with 5 to 10 possible combinations. And at the end, we are waiting for this and that to resolve like when this is like when in the soap opera or in a rom-com, there's the unresolved romantic tension. Will they or won't they get together? So this is the time they spend really finally getting together. It's half of the total time. So, so the total time to go from K to 1 lineages is this. Which is actually more of the same. So now we are adding up the length of these branches. Here from Q to K, we are all adding up those, and this is roughly 1. So so the time to the coalescence finally is 4 Many coalescence times in humans are about 1 billion years. So this means that much of human variation is quite old, much older than our species. Now, adding up, not the distance from the MRCA to the present, but this branch, and this branch, and this branch, and this branch, and this, and this. So we have the total. So this means in this example that we have 1, 2, 3, 4, 5 branches of length T5. We have here to the bottom, and here, and here, and here. 4 branches of length P4, 3 branches of length P3, and 2 branches of length P2. And so this is the sum which comes to this. Now, some applications. Enough with the equations. Let's see how we can apply this. Basic thing that we have to remember that the number of coalescence is a function of the effective population size. The larger the effective population size, the more difficult it is that we start finding coalescences. The smaller the effective population size, they get together quite soon because there's less to choose from. So this is your typical constant size genealogy. These are basically equations that we have seen. But here we have a bottleneck, and the bottleneck means a reduction in effective population size. So in this section here, during the bottleneck, coalescences are easier. You would have in the previous generation fewer ancestors to choose from, fewer ancestors to be derived from. So we see that coalescence are concentrated at the time of the bottleneck. But in expansions, we start not having coalescences, and if we go back in time, an expanding population becomes a contracting population. So now we start with really few coalescences. It's hard. This is an expanding population. Here the population is quite large, and then going back, turning the movie backwards, we get to a situation of a smaller effective population size, and we start seeing coalescences. Coalescences are easy within subpopulations, but more difficult between them. If we have a hard split between two subpopulations, you cannot have a coalescence with someone from the other, with an ancestor from the opposite population. But these are then two small populations, so it's easy to coalesce within and more difficult to coalesce between subpopulations. Here's John temperature drawing the same. Now, let's add mutation. Let's mutations train on top of these genealogies. So, look, mutation 1 will be found only in A. Mutation 3 will be shared by B, C, and D. Mutation 2 will be shared by A, B, C, and D. Mutations 5 and 6 by E and F. Mutation 7 only in F. So we see here these different haplotypes. If two lineages will share as many mutations as they share in the coalescent history. A variant is fixed today if it was present in the MRCA. If it happened before the most recent common ancestor, common to everyone, if these blue mutations here will be fixed now because everyone is a descendant of the MRCA. Any lineage today might be the MRCA of the future. 1 of the 8 billion people alive today, something to be in the future if we haven't killed ourselves, if we still exist, will be the Adam or the Eve of one particular genome section, of one particular genome region. An allele of frequency P. This is why. This is another way of seeing a result that we saw in the first class. That an allele of frequency P that's carried by P people today has a probability of precise the P to become fixed in the future, because there's a probability P that one of those will be the common ancestor of the future. And the average time to fixation of a new mutation is the average time for the MRCA, which is 4NE. In a match of length T, mu T mutations happen because mu is the mutation rate at T is time. We measure mutation rates in mutations in probability per generation. If we multiply this by the number of generations, we get the number of mutations, and this will give rise to these related sites. Then between two alleles that have had 20 generations to accumulate mutations. But this then is it doesn't follow up from this. If two alleles diverge 2 generations ago, like this two, if this is time t, there's the time to accumulate new variants is this branch and this branch, variants that are different between C and D. Let's call phi the average number of differences between two alleles. Then phi is 4n mu. Look, here's they have 2 generations. They for a pair of alleles, T equals 2 and E. Then 2T is 4NE. Given the mutation rate mu, the average number of differences will be 4NE mu. And in a minute we will be calling this theta as well. No, sorry, we're defining these as theta now. This is an important result. We can know from the effective population size and mutation rate we just derived the average number of differences between two lineages. And let S be the number of segregating sites in a sample of n alleles. This will be proportional to the total length of the tree. These n alleles can have the whole tree to accumulate the variance. So this is mu T total, and we remark that T total is 4n times this. So and since we define theta as foreign view, look, theta is here. So S would be equal to theta times this sum. Or theta can also be estimated in this way. So this means that that the average number that we can estimate for nu from the average number of different individuals and from the number of segregating sites. And they should be the same. And if they aren't, something happened. Something happened that violates all the assumptions that we have here. For instance, a constant population size, or for instance, neutrality. So this is the basis of the GMSD test, in which we will be comparing the two estimates of theta, pi, and what observes theta estimator, which comes from the effective the number of segregating sites. So this is you. S comes here, and pi comes there. So if D is positive, if pi is larger than the estimate we get for the number of segregating sites, it means that there's an excess of polymorphism. It means that these alleles that we have, the S alleles that we have, are found at higher frequencies than one would expect under the neutral conditions. So this happened in balancing selection of population subdivision. It means that these alleles are older than they should be, and this happens in balancing selection, which gives which maintains polymorphism or population subdivision. If there's an excess of rare alleles, then the GMSD will be negative, and this is caused in situations in which the terminal branches are long. And so this means that mutations have happened already in the rights of leads to only to one individual. So most alleles will be rare. And we have long terminal branches, as we have seen in population expansions, but also in positive selection. This is constant population size. Pi is equal to theta derived from S. This is the GMSD. In a population, expanding population, pi, this means that the allele frequencies are smaller than one would expect. This is due to the fact that these branches are quite long, and we are accumulating lots and lots of rare variants. And in the population, we have these long early branches, and most sites will be that most mutations will be inherited by this number of individuals or that number of individuals. They will be highly polymorphic. And that was it. That was what I wanted to tell about now. So, any more questions on that? So this is just to justify myself. This just was a size. It went back on me. That's a usual expression when something goes on from smart. Anyway, that was just a taster of the things that you can do if you think in terms of coalescence. I just skimmed the various surface you could have a whole subject to that someone, not me, could teach you 40 hours of coalescence theory, because we could derive many other results. We can have a recombination on top of it. So it's really it was a way of thinking that revolutionized the theoretical population genetics, but not only because it gave the basis for many of the tests that we mere mortals use when analyzing data.
What is working or had to work sometime in life, with the coalescent theory. Everything new, for everyone.
In classes, but well, it's quite hard. Only parent.
Okay, so you had no class. Someone of you? No? Okay, I learned all of this stuff by myself because with the Monod binding lectin gene, there was a hypothesis that there was relative selection acting on, and I wanted to find it out. And I had no one teaching. So this was the best explanation I ever heard.
Mm.
And I love especially the last slide, showing how this phylogenetic tree is not really a phylogenetic university. How this has changed. Problem with Tajima, and also there's also some importantly, is that you can't really differentiate between demographic events and selective.
You're right, absolutely right.
Did this change?
There's a difference. Here is that demography affects the entire genome. The selection is specifically one.
Yes.
So what we do now is, and I will explain more on that tomorrow, that we use the entire distribution of the genotype in the genome to provide sort of a null distribution taking into the data that incorporates demography. So now we think of the genome, a particular genome value as significant if it falls in the extreme of the genome-wide distribution of the genome.
Okay. But to do this, we need whole genome sequencing first.
Whole genome sequencing of a representative sample of humans or of the population of interest. So it's so that's why probably now we have other methods that are more popular, like PBS, and there are really sophisticated methods that probably you don't really need to understand. You press a button and you get your result. There's this relates method that gives you a frequency expected value, frequency trajectory, and on top of that estimates S for you. So it's yeah, because sometimes you have to know how to work with it and we were working with so we had this we are having this collaboration with the biobank in Barcelona. That they have 100 full genomes of people living in Catalonia. So it's not a very good anthropological stuff, but still. And we were looking, so we were looking for an adaptive signals that would be specific to the Spanish population. And so not specific, but the signal that we found was for base persistence. And that student who was working on that used relates to estimate with the base persistent gene, and the result that he got was that it was something like 60,000 years ago. That's 45,000 years before cow. So it's impossible. So yeah, he tinkered with the parameters. Now maybe it's the boundaries of that. Okay, it came that. So it's not. It requires some special knowledge.
Of course.
Some practice and something that sometimes it's not difficult to justify until you get results. Of course, it was a way of calibrating the method. If those parameters get like they persistent right, they probably get all the others fine. So there, what we got was nothing much that wasn't already discovered, but with the with the general European samples in the coalescent project. But yeah, there was something that immediately, if I recall correctly, and then comparing it to MTDNA samples we got that from the Bronze Age. So it's a result that I was interested for political reasons, structural reasons, is that we also did the local ancestry deviation with North African populations. We have 5% of North African ancestry in Spain and Portugal, and now since there is far-right anti-immigration crisis values, it was like, I was hoping to be able to tell people that yes, we are alive because we got this and this and this from North Africa. We got something at the beginning, but it turns out that it's older than that. North Africans also got that from the Bronze Age period.
Okay.
But yeah.
Someone else? No? And then we can close the class. Então, legal que a gente pôde agora da presença. Gente, a teoria da coalescente é revolucionária, né? Né? E adaptar os princípios. E, de fato, só poder visitar.
Tem uma disciplina que às vezes sabe, que é de biologia matemática. Então a gente pega os modelos mais usados e sabe de onde surgiram, de como foram surgidos. Daí parece que isso está
Nossa, eu não sabia da existência dessa disciplina. É biologia?
É saber uma vez.
So you're not taking my picture,
Eu, na verdade, só comecei a fazer isso porque eu só fazia aqui nos Watterson, que era muito mais difícil. Tentei publicar, teve para entendendo? Tentei publicar e todos os servidores, enfim. Você não pode publicar sem fazer sequenciamento dele. E aí fica lá, vem fazer todo o sequenciamento do DNA de toda a região sobre a qual eu queria trabalhar em seleção natural. E aí eu mergulhei nos outros testes. Eu fui entendendo. Eu tinha limite. Isso é um privilégio.
and again, if by some miracle, someone of you is really interested in coalescence, I can recommend as a starting point research using the challenge on coalescence in the right process. Because but most of the cost in there, but you can go deeper.
I like also we use it rather than population genetics.
That's
It's heavier.
yes.
And we use a lot, it's not so much anymore Arlington. Population genetics. So not using it so much. Ok, gente? Thank you.
Muito obrigado.
Então, o pessoal aqui está chamada, que todos já preencheram, né? E o pessoal online também, então. E a gente encerra aqui a aula de hoje. Até amanhã, pessoal. No mesmo link, vocês podem acessar a aula. Tchau, tchau.